because it's important, but I want to get to the muscle, and especially in summer, I want to get to muscle, and I want to get to training, and I want to get to cardiovascular and other stuff. So what I'm going to do with this, I don't know what your comfort level is, but it's going to be like, hey, you guys know this. You know what the central nervous system is. You know what a peripheral nervous system is. You know you have a sensory division that's, senses pain and temperature and all that kind of stuff. And you have motor. I don't know if you guys remember afferent versus efferent. You guys remember that? So afferent, I think, oh, I touched a tack. The message goes, it approaches the central nervous system. So I move my finger away. Well, that motor, it exited the central nervous system. So I just remember afferent, efferent, because you'll see that again. But it's, so review on your own. Another one says review. It's a picture if you like to review central versus peripheral, and you got autonomic. Okay, so just review. When I say review, does that mean I can't ask you a question on the final exam or the midterm? It says, hey, your brain is part of the A, central nervous system, B, peripheral nervous system, C, digestive system, D, reproductive system. I won't. I don't think I'll ask because this is anatomy. So when I say review, it's like very, very low likelihood of any questions being asked. Okay, for your information. Another picture says review, central, peripheral, okay, sympathetic, parasympathetic, that's at the end of this. So you see how fast I'm going. That's how we're going to approach this. Review, relationship. So we've got sensory neurons, we got motor neurons, autonomic nervous system. Okay, I get it. Okay. Structure, review, dendrites, axons, synapses. Okay. Nothing magical, that's anatomy phys. Structure again, picture. This is an older edition of the book, I think. And it says review, so I'm not going to cover it. You don't have to draw it. I won't ask you about axonal hillocks or anything. So, transmission. It says review. I look at this one, I say, okay, it's a cartoon, but it's showing us basically, the synapse. I've got a presynaptic neuron, I've got a postsynaptic neuron. So this guy's going to send a message to this guy. Okay, got it. And then if he gets depolarized, he'll send a message on. It's overly simplified because when we're going to be talking about something called presynaptic potentials. Well, when I see this slide, I say this guy sends the message to that guy. The presynaptic sends it to the postsynaptic. With the nervous system, be careful that just because this guy's sending a message doesn't mean that he's going to be polarized. This guy could be sending part of a message that says, hey, I'm going to excite you. Maybe there's another guy coming along over here that says, I'm going to inhibit you. Another guy coming along over here says, I'm going to excite you. So it's synaptic transmission. Whether or not the postsynaptic gets depolarized depends on the input. And this just shows one input. And I'll clarify this in a second. So neurons are excitable. Okay, fine. Review. Review, I don't do all this potassium, calcium, action potential, repolarization. Yes? So this is getting easy, right? I mean, this is like, review it on your own. I wouldn't spend a whole lot of time on this. I would say you guys have had this in science, biology, anatomy, phys, somewhere where they ask questions about this, about action potentials and this stuff that comes up here. I'm not going to ask questions about it. So I just want you to know it's not because it's not important, it's just because I want to get the exercise stuff, right? And we only have so much time. So you can review that on your own. And you might ask the question, well, if you're not asking questions, why are you even showing them? Well, because they're here. Okay, depolarization, this goes through the steps. Depolarization, repolarization, okay. Yes. Neurotransmitters. Excitatory and inhibitory. So we're talking about neurotransmitters that are being transported across or in the synaptic cleft. It's how we're going to get our information processed. So the close-up of the presynaptic, postsynaptic. What do I want to say about this? It says review. Nothing. Okay. Neurotransmitters, excitatory and inhibitory. It's potential exam stuff, it says. So that means it's not, quote, review. It means it says, hey, Gary, pay attention to this one. excitatory postsynaptic potentials are EPSPs. So if I'm a presynaptic neuron and this is the postsynaptic, and if I'm excitatory, I'm wanting to make this guy depolarize. It's not that he's gonna, but I want to make him depolarize, so I'm gonna send an excitatory postsynaptic potential. Does that make sense? Okay. If I'm going to inhibit him, then I would have an inhibitory transmitter that would be an inhibitory, an IPSP, inhibitory postsynaptic potential. So it causes hyperpolarization. So if I'm sending an inhibitory message, the postsynaptic is less likely to reach threshold. So you go, all right, so I've got excitation and I've got inhibition. All these inputs go into this postsynaptic neuron. How do I get it to depolarize so that it sends the message forward? Well, that's what these represent. Oops. Temporal summation and spatial summation. And there's several slides that come up with this. Imagine this. Imagine here's my presynaptic neuron, and I'm going to try to imagine that that's a postsynaptic neuron. I can, if it's excitatory, I give him an EPSP, and he may reach, it gets closer to depolarization. Temporal means I send him 1, 2, 3, 4, 5 in rapid succession. He's going to be more likely to reach depolarization than if I just gave him 1. So temporal means time. It's repeatedly, quickly giving him information that says depolarize. Spatial is, well, here's an excitatory, here's another excitatory, here's another excitatory. Right? So now I've got 3. It's spatial. It's space. So either increasing the frequency of the information, the EPSP, or the spatial can help that guy depolarize. And the pictures that come up explain it better. This comes out of Lamb's exercise phys book. And I had a transparency, or I don't know, I put red. I tried to show this guy is the postsynaptic, so I wanted to depolarize. So I've got stimulatory are the reds that are going to be making it more likely to depolarize, and then the inhibitory are here. So you look at this and you say, hey, there's inhibitory. There's more inhibitory than theoretically. So it's going to hyperpolarize, right? It's not going to reach threshold. This is threshold. Then I look at this one and I go, okay, there's more of these stimulatory relative to the inhibitory, so I'm getting close, but I still didn't depolarize. And then here it reaches depolarization because there's more stimulation than there is inhibition. So big picture is whether a postsynaptic neuron reaches threshold depends on the relative inhibitory, the EPSP-IPSP ratio kind of thing. And the reason I'm having this as potential exam stuff, because it's fun to say EPSP and IPSP, but also when I was taking this stuff years ago, and maybe you guys are smarter than I was, but people would always talk about nervous input, and they would say, like let's say a motor neuron. I'm jumping into muscle right now. Let's say this is a motor neuron that's going to send. This is a muscle right here, right? And it's going to. So there's my motor neuron. When I send a message down with an alpha motor neuron to the muscle fiber, that muscle fiber contracts always. It just always contracts. So input, contract, input, contract. This is saying, well, kind of input maybe or maybe not reach depolarization, kind of depends. So I always had the nervous system. I always thought that the nervous system, if you sent a presynaptic message to the postsynaptic, it always depolarized, because I was thinking about the motor end plate in the muscle. And it's not. It's just saying kind of depends on the relative IPSP, EPSP stuff. So again, these are going to emphasize the same stuff. It says potential exam stuff. Spatial summation. So basically it's saying I've got excitatory synapses, S1, 2, 3, and 4. So S1 does this, S2 does that, S3 does it. If I add them all together, S1 plus S2, and then S1 plus. So I'm adding all these guys together at the same time, and they cause depolarization. The one on the temporal summation says, let's just look at S1. This guy, and he does it once, he does it once, he does it once, does it once, does it all. Then, again, it's just defining spatial versus, or distinguishing spatial versus temporal summation. A lot of stuff, but it's basically saying whether or not a postsynaptic reaches threshold depends on the relative input, and what you can increase the input is spatial or temporal summation. Okay, this one says potential exam stuff, and it's sensory now. It's saying, okay, we got proprioceptors. Why do I have potential exam stuff? Because I look at this, I say proprioceptors. It tells us just, what our joints are doing, right? Kinesthesia tells us, am I moving quickly or not quickly? What's my body doing? So I think for exercise phys, it makes sense to know what proprioceptors are. It tells me about my body position, tells me what's happening. So it's like anatomy phys, but it's an easy question to ask. Proprioceptors, what do they do? So these are receptors. These are receptors, muscle chemoreceptors. So you say, well, okay, chemo is chemicals. So it'd be nice to have chemical input to my central nervous system about what's happening at the muscle. When I start running up to Williger and I'm making lots of lactate, my pH is going down, my potassium levels are changing, my my PO2, or PCO2. You guys know when I say PO2 or PCO2, it's the partial pressure of oxygen or CO2. Wouldn't it be nice if you guys were making a body, trying to design something that said, when this animal starts running, the CO2 is going to increase, the PCO2, because he's doing metabolism, and we did all that Krebs cycle stuff, so CO2 is going to increase. But it would surely be nice to know when it's increasing a lot. Because if I could have a sensor that would say, hey, PCO2 is really going up, that would be the signal that he's exercising. That, you would think, would say, hey, if PCO2 is going up because he's exercising, let's stimulate breathing. Let's stimulate heart rate. Let's stimulate, let's help this animal. So it makes sense that you would have chemoreceptors that would sense the chemical environment of the exercise. All right. So again, that's just potential exam stuff, proprioceptors, chemoreceptors. Another one, this just emphasizes the proprioceptors. Information about position and joint angle. I won't ask you about free nerve endings, Golgi type receptors, Pacini, I won't ask you these. So whether or not you know that free nerve endings are responsible for touch and pressure, and then Golgi types are ligaments and joint, I don't care. I just want you to know, hey, there's proprioceptors. That's it. That's the potential exam stuff was know that you have proprioceptors. Body position, joint angle. It would be funky to exercise and not know where your body is. I guess does anybody know of anybody that has a disease or something where you don't have this information? Because that would be, I guess if you're underwater and you're
there's this thing where I don't know what it's called, but a person can't feel like half their face and stuff like that, and they just can't feel anything in that area, but they can in the other half. I think it has something to do with the brain because you control the left, the right.
Yeah, I've never thought of this very carefully, but it would be weird to not know. I'm thinking about this stuff and thinking about exercise, and to me it's important when you think about athletes who, I want to say the vestibular apparatus would be important for, ice skaters and gymnasts and that kind of thing, but
you could argue people with really bad coordination, maybe, have really bad
like
body awareness.
Personal trainer kind of stuff. I'm trying to put it in that kind of perspective, like you tell someone, move your arm here, and they don't know what you're talking about. You say, you want to go, come on, because you're trained, you guys know what you're talking about, and then it's like, you got to touch the person, tell them to do whatever.
You see that a lot, with squatting and stuff, like if people have never squatted before, they have no idea, how to move their body in, a balanced position.
That's good. And you guys know it more than I do because you work with clients and stuff, but if you say, Gary, do a squat, I'd probably do it wrong. And then you would say, no, move your back, this way. And I'd say, which way? And, this way? No. Oh, put it over here. Over. So, I guess.
I think babies are still learning proprioception for the first 3 months or so.
Yeah.
So, anyways, it's not a critical, I think muscle chemoreceptors to me make more sense because we're exercising. So it gives the central nervous system something to do about what our hydrogen ion is going up. Yes, he's exercising. Carbon dioxide. Yes, he's exercising. Potassium might not be, readily clear to you guys, but your body, your muscles leak potassium. I mean, that concentration goes up with exercise. So you've got muscle chemoreceptors that sense this stuff. And if you really want to think about it in terms of like big picture, it's if these things are changing, each one of these disturbs homeostasis. And we talked about negative feedback. We said you disturb homeostasis. Your body doesn't want homeostasis to be disturbed. It wants to do something to get it back, negative feedback. So if this is going up and this is going up, you would say, well, that's bad. How do I get rid of it? Well, you breathe. And you breathe, that gets this back to normal. I don't know potassium, but it's nice to have those sensors so that you can deal with the disturbance in homeostasis. And yeah, that's all I'll say about this. And then he's going to say something else, right? One more thing. If I told you there were two people out in the hallway, one person has a really high hydrogen ion concentration and PCO2 is going up more than the other person, I hope you would say that person's probably exercising harder. That's reflexes. Reflexes, review, it says. And you guys know what reflexes are. It doesn't require, conscious thought. So review, there's a couple slides here that'll talk about reflexes, and I'll mention some things that are just review. Reflex arc, I don't know if this came out of our text. It looks like it came out of the text of McCardle. But we're talking, somebody touches pain, there's some sensory information that comes in. What I don't like about this, let's say I was, this is my bicep, so I'm going to touch a flame, and the whole thing is I sense it and I'm going to pull away, right? That's what's going to happen with this. I don't even have to think about it. It just I go, ow, that hurt. When I see this inner neuron here, and it kind of bothers me because I would have rather this picture been drawn with, I get some information, this guy tells the bicep, get out, pull away. The inner neuron and I'm not a neurophysiologist, so I don't know if, how many reflex arcs have inner neurons here, but I think this guy would be there for a different reason than what looks like in this picture. I'll hopefully make it better in a second. I look at this down here and I say, hey, this is the motor nerve, and they call it an alpha motor neuron. Alpha motor neuron. You'll hear me say alpha motor neuron because we're going to talk about something called the gamma motor neuron. But the alpha's the one that says, hey, bicep, contract. Alpha motor neuron says, finger muscle, contract. Flexors, contract. It tells the muscle to contract. Alpha motor neuron. Review. Here's a person touching a tack on the right, on your right. Get some sensory information. And again, this has got an inner neuron here, which it seems weird. To me, it says, touch the tack, information comes, says to this bicep, get out of the way, right? And so he contracts. Here, tendon tap. Tendon says, I tap this, it says stretch. Stretch information comes here. There's no inner neuron, so this makes more sense to me. It says, your muscles being stretched, contracted. Right? You guys remember this? It's yes or no? Yes? It's just reflexes. This is a reciprocal inhibition, which I don't know if you guys remember this. Here's a person touching the tack again, and it says sensory information is going to come, and now there's more stuff here. But what it says is, I touch the tack, and it says to the bicep, get out of the way. Do this. Okay, that makes sense. On the other side, when you think about it, I touch the tack, my bicep is contracting, this is going to relax, right? If it didn't relax, I would if it was a flame, if they both contract, I won't move. So this has got to me, when this information is coming to the bicep that says contract, there's also information coming to the tricep that says don't contract, right? So I do this with the flame on the other side, it's going to be the opposite. So I'm going to do my tricep and inhibit the bicep, right? I'm going to do this. And then one way to think of it is if I stepped on a tack, I would remove my leg, right? I would do this.
Oh, right.
Well, and it wouldn't make sense to do the same thing with this leg because I would, right? So I have to, it's called a cross extensor reflex, and this is just showing reciprocal inhibition, whereas when one muscle is contracting, the other one relaxes. So there's two things going on: reciprocal inhibition and the crossed extensor reflex. It's again, it says potential exam stuff. Probably what I want you to know is reciprocal inhibition is defined as, you know, I don't even think I've ever asked about this. But so just review it. Reflexes, potential exam stuff on somatic motor function. Okay, I look at this slide and I say, all right, I've got peripheral nervous system. I'm going to carry some messages to this muscle. This is the important thing right here on this whole slide, motor unit. The motor unit is the motor neuron, or I could say the alpha motor neuron, and all the muscle fibers that are innervated by it. Right? So, at least in humans, I've got a motor neuron going to my bicep, and that motor neuron is going to touch one fiber, two fibers, three fibers, it depends. So the innervation ratio is how many muscle fibers am I responsible for? If this motor neuron is responsible for 10 muscle fibers, or 100, or 1,000, or 15, that's the innervation ratio. And so you go, okay, motor unit is really the functional thing we're talking about. When I say I want to recruit muscle fibers, I'm on the treadmill and I'm working out harder and harder and harder and harder. I can't just use one muscle fiber, so I want to get, the harder I work, the harder the hill goes and the faster, I want to recruit lots of muscle fibers. Really the way to say it is I want to recruit lots of motor units. So you can imagine I'm on the treadmill, I'm walking nice and easy, so I recruit, I don't know, 10 motor units. I'm just picking a number out because it's easy. And then you say, okay, well now he's increasing the incline. It's harder. Well, let's recruit some more motor units. Okay, instead of 10, let's make it 15, and then 20. And then you say, wait a minute, now it's really getting hard. Let's not only recruit more motor units, let's recruit bigger motor units. So we're talking fast twitch fibers. So motor unit is what we should be talking about. It's the motor neuron and all the muscle fibers innervated by it. My understanding is that in humans, if this is a muscle fiber, there's only one motor nerve connection there. It's not like I get one from this side, one from another side. It's not, this fiber only gets one input. I think in frogs and amphibians it might be a little different, but it's just one motor neuron. Does that make sense to you guys? I emphasize this because I remember when I took a graduate exercise phys class, the professor talked to the class and said, who knows what a motor unit is? And nobody could say what it was. They didn't know. So I thought, oh, it must be important. And it really is when you think about it. This next picture, I think, yeah, here's a picture that's not great. It's illustration of, there's a muscle, right? And you got muscle, these are supposed to be muscle fibers. So I look at this motor neuron, this alpha motor neuron is coming out, and it's touching 1, 2, 3 fibers. So the innervation ratio would be when this guy fires, these 3 contract. Okay, that's fine. This motor neuron is maybe it's weird because it shows like a top and a bottom, but it's going to be intertwined. So this motor neuron touches 1, 2, 3, 4, 5 guys. So when he fires, that's 5 guys that are contracting. So if I wanted a light contract, pick up a paper clip, I can just activate this guy, recruit. If I want to pick up, a backpack, I could recruit this guy. If I want to recruit a backpack or lift a backpack with a lot of weight, I could do both of them. And I look at this and I say, okay, it's a cartoon kind of thing. But do you guys agree that if this guy has 3 fibers, that motor unit is going to be all the same guys? It's going to be all slow twitch, if they're slow twitch, or all fast twitch, because they're all the same guys. This, has 5 guys, 5 muscle fibers, but all of them are going to be, it's not going to be like this one's a slow twitch and that one's a fast twitch, that one's just whatever motor input's going in, it's going to be the same fiber type. So that's why we're talking about motor units, so it's either a fast twitch motor unit or a slow twitch motor unit, even though we talk about fast twitch fibers and slow twitch fibers. Here's, down the road when we talk about, I didn't set my alarm, so let me do that. 10, 20 was the first set, 11, so I'll say 25. Okay. when we talk about muscle, we're going to talk about fast twitch, slow twitch. And the question always comes up, can you change a fast twitch to a slow twitch and a slow twitch to a fast twitch? And that's really a hard question to answer because it depends on what you mean by change. Can you change fiber types? And my answer that I usually give is not really technically, but you can kind of change some of the characteristics from one to another, right? That's kind of you'll see when we do the muscle, it's just a hard thing to answer. One way you can change a fast twitch to a slow twitch, if this is a fast twitch motor neuron, so there's 3 fast twitch fibers, and this is a slow twitch motor neuron with slow twitch muscle fibers, if I cross-innervated, if I took those connections off and cross-innervated, these guys who were, I can't remember which ones I said were fast, these are fast, these are slow, if I switch them, whichever way I said it, the slows will become fast and the fasts will become slow. So we don't do that, you don't do that during exercise, you don't remove the connections and do that, but that's one way you would change muscle fiber type for sure. So that's more basic, more science, kind of experimental stuff. All right, review, vestibular apparatus. I look at this and I say, equilibrium, balance, spinning around on skates, doing gymnastics, doing discus, I don't know, wherever you need some, vestibular input, that's important for exercise, but I don't want to talk about it because I don't know much about it. So review, this is vestibular apparatus using your eyes and your different brain parts, okay. Review what's in the brain. Okay, no, I don't ask questions about this. Review more of brain stuff, motor cortex. There's going to be a slide here where I do say potential exam stuff, and you're going to say, wait a minute, you told us not to know all this stuff, and it's anatomy phys, right? I look at review. Spinal cord's involved. I would say review it. If you're reading the book, they'll just mention that, hey, we talked about the central nervous system, we talked about the brain. Part of the central nervous system is spinal cord, and we know now the spinal cord is involved in movement. So just review it on your own. Here's where it says potential exam stuff, and you go, oh my God, you put the potential exam stuff on the slide with the most information. I'm going to go forward and then go back again. This is saying this, and I look at this. The reason I say it's potential exam stuff is I've asked a question about this. When I first learned it, it was, hey, they talked about the brain and all those different areas, and they said you have a motor cortex. And so my simple sort of thinking was, oh, the motor cortex tells you to move. So your movement starts in the motor cortex, and then you do it. This is saying, oh, no, it's not that simple. There's some subcortical things that do the rough draft. The rough draft gets refined. Motor cortex starts forwarding the message. The spinal cord's involved. Feedback's involved. So if I had to reduce this slide to the most important stuff, it's not as simple as motor cortex says, execute a forehand squash shot. It's subcortical, refining, rough plans, feedback from the muscles. Eventually, I do the movement. And then at the bottom it says, review the last paragraph. So here's the last paragraph from, I don't know which edition it was. Control is very complex. Cooperation in many areas. Recent evidence. So I don't know how recent that was. It was in the textbook. But recent evidence says that the motor cortex doesn't by itself do it. It's motor cortex receives input from a variety of subcortical and cortical structures. CNS, right? And you get the chemoreceptors, the muscle proprioceptors, joint receptors. And then I love this. Much is still unknown about the details, and it's exciting. So I guess I hope it doesn't confuse you that I have potential exam stuff, but I don't really ask you to know all this. I mean, the structures and what happens in every single step. I'm not going to have this box where you have to fill in everything. I want you to appreciate it. So you guys appreciate it? Okay. All right. Potential exam stuff. So when I see this slide, I say, okay, we've talked about the somatic nervous system, has an autonomic nervous system, maintaining homeostasis, not under voluntary control. Okay, it's kind of important. Here's what I think is the important stuff, is you have sympathetic division, parasympathetic division. And whether or not you remember that there's norepinephrine and acetylcholine, I just want you to kind of appreciate there's sympathetic and parasympathetic. When we talk about exercise, if I say parasympathetic stimulation, I generally want you to think, oh, these two divisions do a lot of stuff depending on the organ. You know, it either dilates pupils or causes erections or causes heartbeat or you'll see on the slides that come up, really complex. It's like hormones. When I skip the whole hormone chapter because it's just really complex. So with this, I want you to look at it and say, generally, in general, if I had to put a dollar on the table, I'd say generally excites, generally inhibits. We talk about exercise. If I'm exercising hard and I say there's a sympathetic, response, I'm generally talking about a faster heart rate. If I say, hey, exercise training, I did some exercise training and my resting heart rate went from 72 to 60 because now I'm trained. What's the mechanism? Well, maybe there's more parasympathetic input, more vagus nerve input. That's the kind of stuff I want you. The relative generally excites and generally inhibits. That's what I want you to know. So review, this says review whether you want to know, presynaptic, postsynaptic, and neurotransmitters. I'm not going to cover it. It says review, and I circled slows heartbeat because this was a large 2-page kind of a thing. Parasympathetic was on one side of the page, and then sympathetic was on the other side. And so here's what I want you to know. Parasympathetic generally slows. But you can look at all the different things that it does, and you go, I can't remember all that stuff, right? It's just, so in general, parasympathetic slows heartbeat, accelerates heart rate. So we're talking sympathetic. But it does all the other stuff. And some of this stuff, if I went through every single one, some of them don't seem like they're intuitive. It almost would seem like it's the opposite of what should happen, maybe. I don't know. I'm going over this really fast, which is the signal for don't spend a lot of time on the nervous system when you're studying for the exam. This says review, and it's an old slide from I don't know what edition, an earlier edition. And it said, they were talking about recent evidence shows that, a 5-year study, that exercise is good for your brain. Since this came out a long time ago, it's clear that physical activity in terms of cognitive, function is important. I mean, I don't know if you guys, anybody know about this, about the research? I mean, my impression is that it's, when you think about, hey, old people are having this cognitive decline, and so people are doing crossword puzzles, they're doing Sudokus and all that kind of stuff to keep sharp. This is saying, hey, the physical stuff is just as important as the puzzles you do. And I think there's a really large body of evidence that's saying you should keep moving if you want to maintain your brain health. That was the nervous system in less than half a class period. You guys okay with that? Okay. At least you can say you appreciate the nervous system.
CNS appreciation 101.
All right. What I don't have here, because I didn't think I would cover it this fast. This is like the fastest I've ever covered it, and so I'm hoping you guys aren't freaked out by it. So if I go to winter 16, the next thing we were going to cover was the muscular system. So I haven't had time to look at these slides. Let me look at these.
Are these going to be on D2?
I'll put these, I haven't done it yet because I was thinking maybe, let's see, tomorrow's Wednesday. And you'll see it says, review. It's a lot of the review stuff. Let me not do this just for kicks and stop here for a second and think. Because I'm willing to call it, let's see. We'll get to some muscle, but I don't want to do it today because I haven't looked at it yet. Unless you guys want me to go. I've got 11:07. So the question is, are you guys done?
Well, we're gonna have Thursday to do it, so I think.
So we've got tomorrow is VO2 lab, and that's going to be 5 minutes sitting on a bike, 6 minutes of riding, 7 minutes of recovery. Get the output from the metabolic cart, look at it, and talk about it. Draw a picture on the board. And then if I do decide to lecture after that, if we have time, I can start that tomorrow, and we can do it Thursday. So midterm is going to be on Wednesday, we said, right? So definitely midterm will have chapter control, control systems, stimulus, response, all that kind of stuff. It'll have the bioenergetic stuff. It'll have metabolism stuff, oxygen deficit, oxygen debt, all that kind of stuff. Nervous system and muscular system. That's probably what we'll get through. And so I will try to look for, I've got, I'll try to look for, we can do it.
Are you gonna bring printed out slides, or should we print them out?
Yeah, since I started with D2L, I gave you guys the metabolism slides, but I didn't give you the nervous system slides. So I guess it's your own call. You decide whether you want. I won't bring them. You can either print them out if you want to have hard copies, or if you don't care about hard copies, you don't have to print them out.
So did you say the lab stuff's going to be on that?
The lab stuff's on D2L. I put.
No, on the material.
Oh yeah, good question. Let me look here for just a second. Winter. Shoot. Admin. Midterm study guide. This was winter term. Yeah, I don't know if you guys can see that. It says winter term, topics below. So it says measurement, work power. So there was that one PowerPoint where we talked, I think I've got it posted. I know I gave you guys the handouts. Power output, like how to calculate kilowatt meters per minute, watts, how to convert from liters per minute to mLs per kg per minute. So that's that kind of stuff. Control systems, homeostasis, negative feedback, biological control systems, bioenergetics, that's the whole Krebs cycle stuff, all this. Yeah. Chapter 4, oxygen deficit, oxygen debt, epoch, fuels. And in the winter term, we went to respiration next. So this would, if I posted this, I would get rid of this and I'd put nervous system, muscular system. And then it says anything we've covered in the lab. We did a calibration lab in the winter, but in this class we did Wingate test, oxygen uptake during and after exercise, those two things. So when we do her tomorrow, it'll be oxygen deficit, it's the same kind of thing as metabolism. Wingate is, Wingate test, the anaerobic stuff. Example questions, cycling at 75 RPM on a Monarch 6 set at 3.5 kp would result in, so I go 75 times 6 times 3.5, and I don't remember what the answer is, but you can see it's either this one, or this one says the power out, the same power, does this power output equal the power output of a cycle on a Monarch at 50 using 5.25? So here's a way you got to do it twice to answer, to at least know this one. This says 262.5. Ooh, this one says watts. So whatever number I get when I do this, it's either going to be the same as that number or not. Whatever number I get, let's pretend it's 1575. If I divide this by 6, I'll get watts. So, watts. So you have to know what watts are and what. And this is probably the correct answer. I think this is, if I do it right, 75 times 3.5 times 6 gives me this. And if I take this number and divide it by, if I did this calculation, these two are going to be the same, I bet. And you guys don't want me to do this. You know what I'm at. I'm just trying to give you example questions. Which of the following characterize a rate-limiting enzyme? Is it found at the site near the end? No. Cannot be influenced or moderated? No. Rate-limiting enzymes should be able to be influenced by other factors. Often inhibited by high concentrations of ADP. If I have a rate-limiting enzyme like PFK, is it inhibited by ADP or is it stimulated by ADP?
It should be.
And why?
Because the more ATP that's broken
No,
down,
it says ADP.
But that's a byproduct of ATP production.
The guy in the hallway got his ADP concentrations going up. That means he's exercising because he's using ATP to exercise. So ADP is the byproduct. That should turn on the energy pathways. Right? Okay. So that's not true.
Okay.
So it looks like none of the above would be the answer. Because rate-limiting enzymes are near the top, near the beginning, and that makes sense. Anaerobic glycolysis, this is silly, this is an easy question. Takes place in the mitochondria? No. In the cytoplasm? Yes. During high-intensity exercise? Yeah. Only does I should say fast glycolysis. Does fast glycolysis take place only when O2 is unavailable? Only when there's no. Only is the word that it takes place, when you're working hard, you can still have O2. So it's B and C are true. These aren't maybe not be the best questions, but they were ideas. And I can, if you want me to post this and take off the ventilatory response and then this respiration, I can put, you guys know what we're covering, nervous system and muscular system. But I don't know, I don't find this all that helpful for as a study guide. Maybe you guys do, but students ask me, can we get a study guide? And I say, yeah, we cover chapter 2, chapter 3, chapter 4. And that usually results in growls. That doesn't help us. So, and you guys have any questions about lab write-ups? Yesterday, we took a look at his lab, and I was in the lab this morning looking through research articles trying to find